my name is Jessica Doe. I will be presenting progress towards developing gene therapy to treat amyotrophic lateral sclerosis disease. Amyotrophic lateral sclerosis is a progressive non-degenerative disease that targets the nerve cells in the brain and spinal cord. It affects movements like walking, swallowing, and writing. 90% of ALS cases are sporadic, which means the cause of the disease are unknown. There are currently no cure or treatment for ALS, which substantially prolongs survival. Compared to normal motor neuron, in ALS, the motor neuron loses its contact with the muscle due to different factors such as toxic protein translation or toxic RNA transcription, and thus losing its function. Our research is to see how to keep the motor axon connected to the muscle longer. What is ciliary neurotrophic factor receptor signaling? It protects motor neurons, promoting their natural regeneration, and slows the progression of neurodegenerative diseases such as ALS. The CNTF receptor is activated by CNTF and other related ligands. There are 3 proteins within CNTF receptor: LIFR beta, GP130, and CNTFR alpha. Our previous research found that after cutting the peripheral nerve in the mice, CNTFR alpha increased in the muscle while the nerve is growing back to the muscle. This could be due to muscle operating irregularly and making more CNTFR alpha, or the system is regenerating by making CNTFR alpha to help put itself back together again. To discover the function of CNTF receptor signaling, our research genetically blocked the ligand-binding subunit CNTFR alpha. We noticed with nerve lesion, the axons grew back. The CNTFR increased while axons were regenerating. We also noticed blocking the increase in muscle CNTFR during that time slowed and weakened motor neuron regeneration. In other words, muscle CNTFR plays an important role in protecting and regenerating motor neuron axon. This can be same said about muscle CNTFR that occurs in ALS, therefore may be also helpful in treating the disease. Our previous lab research results also showed that knocking down muscle CNTFR alpha in superoxide dismutase 1 ALS model mice made the disease worse, showing that muscle CNTFR increase is important in inhibiting the disease. The data shown in the bottom left corner underestimated the muscle CNTFR alpha impact since the knockdown left about 20% of the CNTFR alpha RNA still intact. From our discoveries, our studies treated the most widely used ALS model known as SOD1G93A mice with a muscle-trophic adeno-associated virus, CNTFR alpha, which was injected into the muscle well after the mice started to show symptoms. This helped delay end-stage paralysis without side effects. It also increased muscle CNTFR alpha and slowed the disease. Human ALS diagnostics takes about an average of 1 year following the symptoms outbreak, so ALS treatment needed to work well after the symptoms onset. The AAV1 CNTFR alpha effect we found was larger than many other reported for this ALS model, but what's more important was that our treatment was well after the symptoms onset, which none of the other reported SOD1G93A treatments have shown to work. We found that there was another potential treatment against ALS, which was known as cardiotrophin-like cytokine. CLC activates CNTF receptors, and we found that, like CNTFR alpha, CLC RNA is increased in ALS mice muscle. We suggest that increasing CLC RNA may also slow ALS. To test this, we increased CLC with adeno-associated virus gene therapy. As CLC increased with AAV gene therapy, we noticed that the effect is very similar to CNTFR. Again, no other SOD1G93A treatments have been shown to work when starting this late in the disease. In summary, our data suggests that CNTFR alpha and CLC treatments may have substantial clinical impact. Our treatments were able to slow down ALS without side effects in mice when started well after symptoms onset. The treatments can be used before ALS causes are identified and should be able to work with other future targeting of ALS causes. Thank you, and we appreciate your time and participation.